So it's really a pleasure to get to speak to everybody this evening and then also get to share in the cheese tasting that we'll do afterwards. So hopefully you'll still have an appetite for cheese once I'm done. Hopefully your appetite will increase as the talk goes on and not decrease. So I'm going to tell you about the research that my lab has been doing for the past 2 years, trying to develop cheese as a model system for understanding microbial communities. And so I'm going to talk about communities, so I thought I'd just start out with some introduction about what I'm trying to get across here, and that is a community is a group of individuals sharing some common set of interests or a common environment. And the fact is that most microbes in nature exist as part of communities, that is, multiple species living together in a single environment. However, we know incredibly little about what these microbes are doing within the context of multi-species communities. And the reason is because usually what we do as microbiologists is study organisms in isolation in the lab. Okay, so we take E. coli or we take Saccharomyces cerevisiae and we grow it by itself in the lab. And that's very different than what these microbes are experiencing out in the real world. So usually when they're not growing in isolation. They're growing as part of these communities where they're surrounded by many different species. So that is not to say that one of these is necessarily better than the other. You know, we've learned tons about the molecular biology of the cell and how cells function by studying these individual organisms. But there's a whole other sort of world of questions that we can potentially get at about the biology of organisms in the context of communities. So, as I said, in the lab, this has really given us the ability to understand how cells work, but there's questions like how do cells interact with each other? How do they communicate? To what extent are they communicating with each other? And then what determines how a community forms? And the reason that we're interested in microbial communities is because these communities are everywhere, and they impact the health both of pretty much every ecosystem on our planet, as well as the human body. But the challenge here is that most microbial communities are they're not just ubiquitous, but they're also incredibly complicated. So usually we have hundreds or thousands of species all living together within these systems, so very complex. And in many cases, we don't actually know how to culture these organisms in the laboratory, so it makes it very difficult to study. So just as we use E. coli or Saccharomyces as model systems for understanding how cells work, can we come up with model systems for understanding how microbial communities work? And so some of the criteria that we were looking for in a model systems is that it would be simplified in a number of ways compared to these more complicated ecosystems, that we would be able to reproduce it in the lab. Hopefully it's accessible so we can easily get samples of our system. And then ideally we want to be able to manipulate it at many different levels so that we can understand how it works. So whether we want to understand which individual genes are contributing to the functions of the community, which individual species are important. We can manipulate the community as a whole or what they're growing on, and then the environmental parameters in which the community is growing. So those are all things that we're hoping to find in a system. And so I spent a few months sort of trying to figure out what sort of system might represent these criteria that would make it interesting to study. And I was really interested in food and food science, just because I love to eat. And learning a lot about food science, what I realized is that fermented foods are actually an example of a system that meets a lot of these criteria. So these are microbiologically rich and interesting systems, but they also are relatively simple compared to many other microbial systems. So this slide just illustrates that pretty much every culture on the planet has some form of fermented foods. Everything from dill pickles that we all eat on burgers, and to cheese, beer, sausages, things like other fermented vegetables like kimchi, things like soy sauce is fermented, even things as common as coffee and chocolate are examples of fermented foods. So these are all things where microbes play a role in the production and preservation of these particular foods. So these could all be examples of systems that we could study and learn about the microbiology of communities. And of course, the system that I chose to focus on is cheese. And I think cheese is one of the most interesting fermented foods because of the diversity of styles and flavors and smells that are present, and those all represent diverse microbial functions. And so I thought I'd give just a brief introduction to cheesemaking, just so we all are familiar with what I'm talking about when I say cheese and why microbes are important in cheese. So all cheese starts with milk. Okay, so this is usually cow milk or goat milk or sheep milk. And then this milk is fermented and coagulated. So lactic acid bacteria like Lactococcus are added to the milk. And if you just add Lactococcus to milk and let that ferment, does anybody know what you'll get? Yogurt, right? So the live active cultures in a can of yogurt, those are Lactococcus or Lactobacillus species. They basically ferment the lactose to lactic acid to produce energy for their growth, and the acid, the lactic acid acidifies the milk, starts to cause the proteins in the milk to coagulate. So sort of if you were to squeeze lemon juice in a glass of milk, it would curdle, right? So there's a similar type of process happening in milk during cheese making. Now the difference between yogurt and cheese is the addition of rennet. So rennet is an enzyme that it's actually a protease that is specific for the protein in milk, which is called casein. And so in addition to the activity of the acid starting to coagulate the casein protein, the rennet also is starting to chop it apart and cause it to coagulate as well. So you end up with a much firmer curd than you do with yogurt. And that curd is the starting point for all cheeses. So this is just a picture of some fresh curds that were cut into cubes. And when you cut the curd, you release the whey. Okay, so you start with milk, you end up with curds and whey, and that's that's the beginning of all cheese. And the interesting part in my mind is what happens next, and that is how do you go from this very simple starting product of fresh curds, which don't really taste like anything; they just taste like milk, basically a little sour. And then somehow you end up with hundreds of varieties of cheese. They all look different, they smell different, they taste different, they have different textures. It's just completely amazing, the diversity that you have in the world of cheese. And the way that you get to this diversity are through two main manipulations. The first is through what we as humans do to manipulate the curds during the cheese making and aging process. So you can do things like cook the curds or press them, to get moisture out. You can salt the curds, and then you can age them for different amounts of times. And this is all going to have different effects on the cheese. And really, actually, a lot of what we do as humans, as cheesemakers, is are changing the cheese in ways that will change the microbial environment and change the types of microbes that are present that will grow on the cheese. So the second component to this diversity is really the manipulation that's being carried out by the microbes. So there's microbes inside the cheese, the lactic acid bacteria, and then there's a whole suite of microbes on the surface of the cheese, which is what I'm going to spend the rest of the time talking to you about. And they're all growing on the cheese and making a living off the cheese. So they're consuming the fat and protein and sugar that's in the cheese and transforming that into many different compounds that are important for the flavor and texture. And so I thought I'd just give you guys a brief glimpse of some of the things that microbes do that make cheese smell the way it does. So these are 6 molecules that are known to be produced by microbes during the production of cheese. Some of them seem like they would be a good thing, like maybe this 2-methylpropanol has the smell of bananas. That's not too bad. Methanethiol, that one smells like rotting cabbage. Methionol, this smells like sort of roasting meat. Butyric acid smells like sweaty gym socks. Diacetyl is the smell of butter and the taste of butter. And this compound here, benzaldehyde, smells like bitter almonds. So the way I like to think about this is that there's different microbes growing in the cheese. They're all carrying out their own metabolism, producing these different compounds. And in whatever combination you have those microbes, you have this combination of interesting molecules involved in the smell of the cheese. So depending on the microbial community, you're going to have different amounts of these molecules, and you're going to end up with a different taste and smell in the cheese. Okay, so in maybe small amounts of butyric acid, that might actually lend an interesting characteristic. And you guys will probably taste cheese today, or you may have tasted cheese in the past that are really funky smelling, very stinky. They have lots of bacteria that produce molecules like butyric acid. And some people enjoy that, and other people don't. It's sort of an acquired taste. But this is sort of one of the reasons why we have so much diversity in cheeses. And so the part of the cheese that my lab is the most interested in studying is the rind of the cheese. And these are just some close-up shots. This is the surface of the cheese. And we think that the rind of cheese is not only interesting in terms of the aging of cheese, which is sort of what cheesemakers are interested in, but we think that this actually could be a really nice model ecosystem for studying microbial communities. So the rind is actually a surface-associated microbial community. So this is a slice of cheese that I put under a microscope. And this is just a couple millimeters thick. And what this is it's actually a microbial biofilm. So it's a community of microbes, and it's actually both bacteria and fungi living together, growing as a community on the surface. And I like to make the analogy to other surface-associated communities, such as this one from a salt marsh that you might find down on the Cape, where you have these mats of microbes living together. And these types of communities go back in the fossil record for billions of years. So this type of lifestyle of microbes growing on a surface as a community is really an ancient mode of existence, and something you find in many different environments, not just cheese. And this is a close-up of the rind. So this is a scanning electron micrograph of the surface of a cheese. And you can see this is the surface here. This is the top of the biofilm. And this is starting to crack open. You can start to see some of the microbial cells. And then if you look closer, if you just sort of imagine shrinking yourself down to the size of a microbe and standing in the middle of a cheese rind and look around, this is what you would see. So this is, if you guys can see this, there are some fungal spores here. And then this like big clumps of bacterial cells. And you can see different shapes and sizes of bacteria. So these are different species all living together, really in really close contact within this community. Yeah? Are there archaeas there also, plus bacteria? So we think there could be a small proportion of archaea. There have been some methanogens found in fermented foods as well, and cheese as well. But it's dominated by bacteria and fungi, based on the studies that we have done so far. And one way you can tell that there are many different species living there together is if you just scrape the surface of the cheese, bring it back to the lab, plate it out on just some basic lab media like LB. I think most of these were actually just LB that I had hanging out in the lab. And I plated them out, and you see these beautiful colonies growing up. And these are 4 different cheeses, and you have 4 different types of communities. And you can tell that just based on colony morphology. And this community forms over time during the aging of the cheese. So this is one example where you have 3 batches of cheese at different stages of aging. So this batch here is only a few days old. It basically looks and tastes like solid milk. Not a whole lot going on there. And then a few weeks later, what you can see is the surface starts to become pigmented. And these are the microbes starting to colonize and grow on the surface of the cheese. And then this is about 2 months of aging, and you see this fully formed community on the surface of the cheese. Yes?
Where do those bacteria come from? Are they always there in tiny amounts, or do they
Yeah, so it's a great question. So I told you the lactic acid bacteria are inoculated into the milk. But these bacteria, in many cases, are not inoculated. So they're coming in from the environment somewhere, either from the milk or from the cave, from the cheesemaking facility somewhere, but we don't exactly know where. But I think it is really remarkable that we have such reproducibility, wheel to wheel and batch to batch and year after year of these same communities forming in a reproducible way, even though they're not being inoculated in any sort of standardized way. So the reason we think that this particular microbial community is interesting as a model system, one is that we have many replicates of it. So you can just see here, within a single batch, you have about 100 different experiments. And these cheeses get made over and over again. They're reproducible. They're easy to access. So we work a lot with cheesemakers in Vermont. Or we just go to Formaggio Kitchen to get samples, which you guys will taste some cheeses from there after. The system can be manipulated, and I'll tell you more about that, but I sort of think of all of these different types of cheeses as manipulations of this community that are already in existence that cheesemakers have figured out how to do. So we actually might be able to learn something about how to manipulate microbial communities just by studying the different manipulations that people have already carried out. And then one really wonderful thing, which I'll spend a little bit more time talking about later, is that we can follow this system over time very easily. So this is something that can be challenging in the study of microbial communities is it's often difficult to say when the beginning and end of a community is and get samples evenly over time. And this is something that we can do really nicely with the cheese. Yeah?
I'm a cheesophile, fromageophile. And my question is, the rind is on the outside,
Yeah.
but the flavor's on the inside. And if you cut the rind off the cheese, you still have all that flavor. What's the process by which the flavor gets from the rind to the cheese? Or is there a whole different microbial community doing its thing inside the cheese?
So there's the microbial community inside the cheese that are the predominantly lactic acid bacteria, so not very diverse, but they are carrying out some metabolism inside the cheese. And then we have this whole suite of organisms on the surface, and what's happening is they're metabolizing the cheese from the outside in. So they're secreting enzymes to break down the cheese. So if you eat a cheese like a brie or camembert, often you'll notice that right under the surface, you see it's kind of gooey. That's where it ripens first. And that's because it's ripening from the outside due to the action of the enzymes that are diffusing from the organisms on the rind.
And that's what's changing the flavor.
Yeah, so the enzymes will diffuse in, and also the metabolites from the organisms will diffuse into the cheese and impart a flavor. So often if you eat a cheese, the stronger flavor is right underneath the rind compared to the middle of the cheese. Yeah, but it depends a lot on the type of cheese that you're looking at, how much impact the rind will have on the flavor. Okay, so what our goal is in studying the system is really to develop this as an experimental model system, something where we can study the microbial community in its natural state, so that we can make observations about the ecology of the microbes and generate hypotheses about what might be determining how the communities form or what the interactions are between the species. And then what we hope to do is be able to pull the system apart completely, so have these different species in isolation in the lab. And what that will allow us to do is actually test hypotheses about microbial ecology, which is something that's been really a challenge in many microbial systems because it's hard to grow the organisms in the lab and it's hard to reproduce the environment that they're growing in the lab so that you can actually test your ideas about what's happening in the system. So we want to be able to do hypothesis testing in lab so that we can eventually get down to mechanisms of what's happening. Okay, so I'm going to talk about two main types of experiments that we've been doing over the past 2 years. One is that we've been spending a lot of time just trying to characterize the system. So it's kind of amazing how little we know about the food that we eat. And especially for fermented foods, there really haven't been, there hasn't been nearly enough study of the microbiology of these systems, and we don't really have a clear picture of what these communities actually look like. So what we've been doing is collecting samples from as many different cheeses as we can get our hands on. So we ended up with about 165 different types of cheese from 11 different countries. And we had triplicate wheels for each of these types of cheese so we can look at reproducibility within our samples. So we got 3 different wheels for each one. And we tried to sample across the 3 major styles of cheese. So we have washed rind cheeses, which during the aging process are washed with a brine solution, so a salty solution. So this creates a very moist and salty environment on the surface of the cheese. The rinds look very distinctive. They have this reddish-orange characteristic. And they have this very stinky smell to them. So these are the really funky cheeses that sort of smell like feet. And it was thought that this reddish-orange pigment was determined by a specific bacterium called Brevibacterium linens. So if you ask anybody in the cheese world what the reddish-orange pigment is on the surface of these cheeses, they'll tell you B. linens. And B. linens is known to impart a really funky aroma on cheeses because it's responsible for a lot of protein breakdown. And the protein breakdown, it does it to the point where it creates a lot of volatile sulfur molecules. So things like the butyric, the methane thiol type compounds. So this is one group we wanted to look at the microbiology and see if it actually was Brevibacterium linens or what other members might be present in this community. The natural rind cheeses, these cheeses are produced and then just left to sit in the cave. And there's not really, they don't really do a whole lot with these cheeses, so it's very natural colonization and no disturbance. And then the last category are the Bloomy rind cheeses, which are the one example where they are inoculated, and they're usually inoculated fairly heavily with the spores of certain filamentous fungi. And so these are the cheeses like Brie and Camembert, which have this very fluffy white surface on them. So we collected samples across these 3 styles, and we wanted to ask how different are the microbial communities in these different cheeses within a style or across the styles. And we're collecting data on the environment because we want to know which factors have the most impact on the structure of these communities. So we're looking at pH, salt, moisture, as well as geography. And we're also interested in seeing if we can detect patterns between the species that might suggest that certain organisms are interacting with each other. So if you always find one species in a system and not the other, that might suggest some sort of negative interaction, or if you always find them together, that might suggest a positive interaction to generate hypotheses that we could actually test in the lab. Yeah?
Did you just use cow's milk cheeses, or did you use mixtures of cow, sheep?
We have cow, sheep, goat. We have raw milk, pasteurized milk. And those were the, I think, besides these 3 categories and across geographical locations, so the parameters that we tried to sample. Okay, so I thought I'd go briefly through just how we did the sampling, although I think you guys have heard a lot about genomics already, so I'll go through this quickly. We collected samples from the surface, we scraped them, put them in tubes, brought them back to the lab. We extracted DNA. We PCR amplified the ribosomal marker genes for both the bacterial community and the fungal community. So for the bacteria, it's the 16S. For the fungi, it's the ITS. And these are both just sort of the fingerprinting genes that we can use to compare the species to each other. We used next generation Illumina sequencing. And this gave us many millions of reads, and we matched these to databases to do our analysis. I'm going to just show you the bacterial data. We have now actually data from the fungal portion of the community too, which is really exciting because there have been very few studies that have looked at both bacterial and fungal communities together. But I think we're just going to have time for the bacteria today. So this is the data. What this is a representation of each of the individual communities from each of these 165 different cheeses. And the cheeses are along the x-axis. And along the y-axis is the relative abundance of the species within the community. Okay, so the way to read this is that these sets of communities here are dominated by this red species, which is Lactococcus, whereas these communities here along this set of natural rind cheeses have decreasing amounts of this Brevibacterium. And so I've just grouped these here by the 3 different styles of rind. So these are the cheeses that we sampled from the Bloomy rind category, from the natural rind category, and from the washed rind category. And I'll go through in a little bit more detail some of the interesting observations that we had, and we're really working hard to analyze this data now to try and generate some hypotheses that we can test. One thing you can look at is this question of, is this actually a simpler system than other environments? And I said in other environments we have hundreds or thousands of species. Here we're on the order of 10 or so species that make up the dominant organisms. So we have around 7 in the Bloomy natural, 8 in the natural, and 10 in the washed rind cheeses. So what are some of the highlights of this data? One of the highlights is this idea that most of these organisms are not inoculated into the cheese. Okay, so I've just highlighted the things that we know are starter cultures. So these are the lactic acid bacteria that we're actually picking up in some of the rind samples in the Bloomy rind cheeses. So these are all Lactococcus and Streptococcus, and we see them in a few other samples, but fairly infrequently. And all of this black area is sort of the black box of things that we really don't know where they're coming from and really what they're doing. So the majority of the organisms that are on the surface of the cheese are not inoculated there in a controlled sort of way, and we really have very little knowledge about what these organisms are actually doing or contributing to the cheese. So one interesting observation is that Brevibacterium, so I was telling you about the Brevibacterium linens that everybody thinks is the dominant microbe in the washed rind cheeses. So this is the washed rind set of cheeses. We see Brevibacterium, but it's definitely not the dominant member of the community in most cases. And actually, it seems to be dominated in many of the natural rind cheeses. So this is sort of interesting. We don't really know. We know these cheeses are stinky, but we don't know what's making them stinky if it's not Brevibacterium. And why isn't there as much Brevibacterium as people thought there were? And what is Brevibacterium actually doing in the natural rind cheeses where we didn't really think that it was an organism of interest? Yeah?
The natural rind cheeses aren't as sticky either.
No, they're not, exactly. So it doesn't really make sense.
What are you using as a washing? So you say washing.
So the washing is usually a salt water solution. Yeah.
Well, that have anything to do with the
That
less being seen.
Yeah. So let me get back to that in one second. We'll talk about the washing. So with another interesting thing is that we have a lot of Staphylococcus species. So when you hear Staphylococcus, you may think of Staph aureus, which is a really nasty pathogen. So this may seem concerning that there's staph in cheese, but these are actually non-pathogenic types of staph that are found everywhere in the environment, growing on the surfaces of our skin and the surfaces of plants, and they're pretty much everywhere. And they seem to be a really important member of at least the natural rind community of cheeses. So this is one group of organisms that we're really interested in understanding more about their role in the communities. And then the other thing is back to this washing question, that what we're finding a lot in the washed rind cheeses are salt-tolerant bacteria. And cheese in general is a salty environment, so we're seeing them in other types of cheeses as well. But in particular, the washed rind cheeses, because they're being constantly washed with the salt water, like salty solution, we see many different halophilic organisms. So in particular, we have a lot of Halomonas, and we have a few other things like Vibrio, which is, this is Vibrio casei, so it's related to Vibrio species you would find in the ocean. In fact, these 4 here are all closely related to organisms you would find in the ocean. So we're not really sure how they're getting there, if they're maybe coming in from the salt, or if it's just that this environment is very similar to the saltiness of the ocean water, and that's why they're showing up. Yeah.
Are your cheeses aligned in a continuum, or are they just randomly placed on the x-axis? Because you have these nice patterns. I just wondered if there's anything about them that...
Yeah. So I just sorted them based on the abundance of the dominant organism within that group. Yeah. So the dominant organism in the wash rind is Halomonas. So these are, these communities here are almost entirely Halomonas. Yeah. Yes.
That being the case, on the far left of the natural rind group, that's
Yes.
where you had the most, was it Brevibacterium?
Brevibacterium.
Was that a particular stinky cheese compared to the others? Or?
I don't know, although we do have a lot of samples remaining of our little cheeses, and we're thinking of doing some volatile analysis for those samples, but it's not something we've actually done so far. Okay, so that's looking at the final communities, and we're just sort of getting going with the analysis of that data. But these data were only the endpoints of aging, so a single snapshot in the life of this community. And as I said before, one of the nice things about cheese is that we can follow the community formation over time. And so what we did is we set up a study at Jasper Hill Farm in Vermont to study the aging of one natural rind cheese. This is Bailey Hazen Blue, and it undergoes about a 2-month aging period. So this is fresh cheese, and these are about 1 or so week intervals down the aging period. So what's amazing is that you can see the changes in the microbial community by eye. So we knew there were interesting things happening, but we wanted to understand what these profiles actually look like. So we went in, collected samples, and sequenced these. And so these are, what you're looking at now is a similar type of data where we're looking at relative abundance of species within a sample. And these are triplicate wheels. So we did this for 3 individual wheels within a batch, and we did this for 3 batches. So this is just 1 batch of cheese. And we have, in this case, both the bacterial and fungal data. And what you see is this really beautiful succession of organisms over time. So we start out with the lactic acid bacteria. This is pretty much just the curd that we're sampling at that point at 0 days. So this is a fresh cheese. And then a week later, what you can see is that the community starts to be taken over by these Staphylococcus species that I mentioned earlier. And so the staph dominates the community for a few weeks, and then you see this Brevibacterium start to come in and eventually dominate the community. So this raises the question of, what is the relationship between these 2 organisms? Are they interacting in some way? Is it the case that Brevibacterium requires staph to be present first in order for it to grow? And we can make a similar set of observations with the fungi. We have the succession also happening, and interestingly, it sort of transitions in a similar time frame to a community that's dominated by a yeast called Candida catenulata, to one that's dominated by mostly 2 different filamentous fungi. This is a penicillium species, and this is a fungus called Scopulariopsis, about which we know very little. And what's nice about this is because these are filamentous fungi, we can actually see them on the surface of the cheese, and this is exactly when the cheese starts to become fuzzy. We start to pick up these organisms from the surface. So we're really interested in knowing then what causes these changes over time, because this will give us some insight into how these communities are developing. And one of the great things about the cheese system is that we actually can culture pretty much everything from these communities. So this is just a list of some of the bacteria and fungi that we have isolated and we have in a culture collection in the lab. And what we've shown is that for everything that we find on the cheese that's at least 1% or greater of the community, we actually can culture it in isolation in the lab. And what that allows us to do is then try and test these hypotheses about what is determining the community formation. So we can mix together different species and ask, can we reconstruct the communities in the lab, and can we then dissect them to understand what's happening? And so we're doing just that. So this is one of these first types of experiments where we're looking at this succession of the Staphylococcus and Brevibacterium that I just showed you from that natural rind cheese. We take these two species, we mix them together in equal number on a cheese curd agar plate. So this is our in vitro cheese. And we just take cheese, we freeze dry it, we grind it up and mix it in to agar plates. So we have 10% cheese. And so we plate these bacteria on the surface to mimic the rind, and we just incubate them for ends up being a total of 2 weeks, and we sample over that time course to see if we're seeing the same pattern. And this is the data we have so far. So this is the actual cheese that I showed you, the succession that's happening with Staph and Brevibacterium. And this is what we can see in the lab. So these are our spots of our little mini rinds. And what we see is the same pattern of succession. So the early community is dominated by Staphylococcus. And remember, we inoculated these at equal levels at the same time in the initial community. Yet the Staph is dominating early, but it's the transition happens to eventually be dominated by Brevibacterium. So we're now trying to pull this pattern apart in the lab and ask, is Brevibacterium actually stimulated by Staphylococcus? And we have some experiments from this past week that show that it is really strongly stimulated by Staph, which is really exciting to us. Okay, so I think this sort of what I hope to have gotten across is that these communities do represent a system which is simple, culturable, and experimentally tractable. We've started to make some observations about the ecology and generate hypotheses from those. We've pulled it apart. We have all these organisms in culture, and now we have actually showed we can start to reconstruct these communities and test hypotheses in the lab. Okay, we have a few minutes left. And so what I thought I would do is tell you guys just a little bit about the cheeses you're going to taste. And we don't have a whole lot of time, so I'll go through this really quickly, and I only have a few examples, so there'll be many more cheeses present. So one of these cheeses is called Boucheron. This cheese is from France. It's made from goat's milk, and it falls in this Bloomy rind category. Okay, so it has this fluffy white surface. And the dominant organism in these types of cheeses, there's two dominant ones, is Penicillium camemberti. This is a scanning EM of Penicillium candida. It's named for paintbrush. So Penicillium, the structure of the spores look like a paintbrush. And this is what it looks like growing in the lab. It's an aerobe, so it only grows on the surface of the cheese. And it secretes proteases into the curd, so these diffuse into the curd. They break down the protein, producing peptides, which have this sort of mushroomy flavor, and they impart the gooey texture of this type of cheese. And the other fungus that is important is Geotricum candidum, and it's really interesting because it has a lot of diversity in different strains, so you can find different types of Geotricum growing in different regions, and they have very different morphologies and metabolic properties, which would impart different characteristics to the cheese. And what Geotricum does is it secretes lipases into the cheese, producing free fatty acids, and eventually breaking them down further to methyl ketones and thioesters. So often when you eat a cheese with Geotricum, it has a very cabbagey kind of smell, so things like these methane thiol compounds. The next kind of cheese is a blue cheese. So you guys are going to have, I think, two examples of a Gorgonzola. One, Gorgonzola Dolce, which is creamier and younger, and the Gorgonzola Picante, which is older. These are made in Italy. And the way that blue cheeses are made is that they're actually inoculated with the spores of this blue fungus called Penicillium roqueforti into the milk. And when the cheese is made, this particular fungus will only grow in microaerophilic conditions, so it doesn't like a lot of oxygen, but it can't grow without oxygen. So the cheesemaker will actually spike the cheese to let in just a little bit of air into the pockets of the cheese, and then the fungus flourishes in that environment. And this is a picture of Penicillium roqueforti. And what it does is it, again, secretes specific lipases, breaking down fats into free fatty acids. One of these free fatty acids has the peppery flavor that you associate with blue cheese. So often blue cheese is spicy, and that's because of free fatty acids from the breakdown of fats by this fungus. And then these fats are further broken down to these smaller volatile molecules, one of which is 2-heptanone, which is the characteristic aroma of blue cheese. So if you eat blue cheese, you know it's blue cheese. It smells like all blue cheeses sort of have a similar smell, and that's because they all have this compound that's produced by this fungus. And then the last one I thought I'd tell you is about alpine cheeses. This is my favorite. We have one example of alpine cheese. It doesn't have a whole lot of holes in it, but it might have some of the characteristics. So Brevibacterium is made in the Pyrenees of France, and I think some people might think that these cute little mice are responsible for the holes in Swiss cheese, but actually it's a bacterium. It's called Propionibacterium. It's an anaerobe, so it only grows inside the cheese after the cheese is made, so there's very little oxygen. It starts to grow, and what it can do is that it uses the leftover lactic acid from the fermentation, initial fermentation, and it ferments the lactic acid to propionic acid, which is the characteristic flavor of Swiss cheese, the sort of like tangy flavor. Also acetic acid, and CO2, which is the key thing. So what you can imagine is that everywhere there was a Propionibacteria growing, started growing, producing CO2, and created this bubble around itself in the cheese. So, okay, I just want to thank my lab, Ben Wolf and Julie Button, who are 2 postdocs in my lab, and we go up to, this is up at Jasper Hill Farm, where we're collecting some samples and reagents. Marcella, we had a visiting PhD student who did her PhD thesis on Parmigiano Reggiano. And this is the caves at Jasper Hill Farm, where we sample a lot of our cheeses. Our lab is in the 4th floor of the Northwest building, and we're part of the Bauer Fellows Program and the Center for Systems Biology. And we get a lot of support from Peter Turnbull, who you guys may have heard talk about human gut microbiome, and the Bauer Core facility that does a lot of the sequencing work for us. So I'm happy to take questions. So yeah, go ahead.
You said that you had found some relationship between the Staph aureus and the Brevibacterium. So how one stimulates the other? Can you tell us a little more about that?
So it's not Staph aureus, just to be clear. So we just have started doing these experiments in the lab, and Julie is doing these experiments. She's the one doing all the reconstructing the communities, and she just showed me the results like 2 days ago. So we don't know what the mechanism is. We have a few ideas that we're testing, but what we can see is that if you try growing Brevibacterium alone, it grows very slowly in a cheese curd medium, but if you add staph to it, it grows much more quickly. So that's the basic result.
Is there some metabolic connection?
So that's what we're interested in figuring out. Yeah, we don't know. Yeah.
I have 2 questions about the Brevibacterium being stimulated by staph. Could that be used to either diagnose staph, a staph infection, a bad staph infection, or possibly as a treatment? If people have a staph infection, we give them this Brevibacterium, which is harmless, and it might help slow down the growth of the staph.
So could we use this as a possible like probiotic type?
I mean, obviously, in addition to antibiotics, but or for diagnostic purposes. You know.
Yeah, I don't know. I can imagine that in some cases, if you have certain staph species which are non-pathogenic, you might be able to outcompete pathogenic staph. But yeah.
Or the Brevibacterium could outcompete this, or partially outcompete the staph and help the antibiotics be more effective. I don't know.
Yeah, it's an interesting idea. I don't know.
Crossed my mind.
The question was whether it might be a clinically useful tool if you were able to maybe, if you have a staph infection, to add Brevibacterium, which would maybe take over the community, or if it could be used for diagnostics. Yeah, that was a question.
So you talked about the bacteria communities that form on the outer parts of the cheeses, and it's not really clear where they come from.
Right.
Have you had time to look at the cheese makers in different regions to see if there's a correlation between them and their cheeses?
So, so it's definitely a possibility that some of the microbes are coming from skin, because these are microbes that are sometimes found on skin and are closely related to organisms found on skin. We think it's probably more likely that they're coming from the skin of the animals. So during the milking process, for example, from the cows or the goats or sheep. But we haven't done any swabbing of environments or of cheesemakers. Yeah.
Is there a change in pH as the cheese ages and the dominant bacteria colonies change?
Absolutely. So I didn't talk about this, but there's a pH change is one of the big environmental changes that's happening during the aging of a cheese. So it's thought that the early colonizers deacidify the surface of the cheese, which might allow the growth of later organisms. There are also changes in salt because usually cheese is salted on the surface, and that will slowly soak into the cheese and change the concentration of salt over time. And then there's changes in moisture over time as well as the cheese is sort of drying out in the cave environment. Yeah, so we're also, besides just looking at the interactions that are driving the community, what are the environmental changes that might also explain some of the changes?
I got two. One of them is about bouchon specifically, it's one of my favorites. And that is, when you buy bouchon, the layer of, if you like, digested cheese is about 1/4 inch thick.
Okay.
Right? Do they stop it at that stage, or is that just where the enzymes stop going? Because it's like a chèvre in the middle, and then that nice gooey, flavory stuff on the outside.
So the question is, why for a cheese like boucheron is the ripening limited to the outer layer? And it's because the cheese is only a few weeks or a month or so old. And if you take that cheese and keep it on your shelf at home, which you can try, that layer will migrate in further and further in, and eventually the whole cheese will sort of start to liquefy.
Oh, I like the best.
Yeah. You can age it at home.
I will. The problem is it never lasts that long. The second question is, in this country, we require that cheeses be pasteurized, and to me, we're losing a lot. We may be taking a little risk, but everything worthwhile is risky, right?
Yeah.
But we're losing a lot in that there are a lot of very flavorful cheeses we just can't get off there yet. Really sad.
So, so the regulation for cheese. So the question is, what about raw milk cheeses, and are we losing a lot of diversity and flavors by not having access to raw milk cheeses? And in the United States, we actually do have access to raw milk cheeses, but they have to be aged for 60 days or more. Okay, so you can't have a raw milk camembert or boucheron, for example, because they are not aged long enough. But for a lot of natural rind cheeses, there are many cheesemakers in the U.S. who are making raw milk cheeses, and there's imports of European raw milk cheeses. And there hasn't been very much research, that really demonstrates, I think, very clearly the connection between increased diversity from raw milk cheeses and increase in flavor or aroma. But I imagine that to be the case. And we know that raw milk cheeses, even from our data, have more diversity than pasteurized cheeses in terms of the total number of species that we see. So I guess my stance is that I think the microbes are incredibly important for the flavor, and we know that microbial diversity equals more diversity of flavors, at least in theory. So I would suspect that raw milk cheeses would be more interesting, although I've tasted a lot of raw milk cheeses, a lot of pasteurized cheeses, and it's not necessarily the case that raw milk cheeses are superior to pasteurized cheeses. I think it just depends on the cheese and what you're looking for. Yeah.
Yeah, are people interested in engineering new cheeses? Like, can they take members from one community and try to introduce them into other communities and get novel cheeses and novel? Are they interested? Do they do that? Or is that
So,
nature?
I mean, cheesemakers have been doing that for a long time. I think trying to change the cheese in different ways to promote, to come up with new types of cheese. And that's part of why we have so many different types of cheese, because people have been successful at doing that sort of thing. There are a lot of cultures you can buy that people will add to cheeses, although what we're finding is that it doesn't look like, in most cases, those cultures will actually end up on the cheese. It's actually the whatever the native microbes were that dominate the cheese. So I'm sure there's interest in doing that, but maybe some of the research that we're doing might shed light on more successful ways to do it, or
Seems like you're
different ways
kind
to
of
do
moving
it.
in that direction by dissecting out the various
Yeah,
members.
exactly.
You showed very nicely the separation of all the cheeses versus the dominant bacteria. Have you noticed any patterns or correlations with the bacteria that you would see, or dominant bacteria see, based on regions of where you isolate those cheeses? So in other words, cheese from France, they have a certain type of bacteria versus...
Right. Yeah, so that's one thing we're interested in looking at, whether there was some geographical basis of cheese. So you can imagine that if you make a cheese in France versus make it in Vermont, you might end up with very different tasting cheese, even if you made it the same way. But what we're finding is that geography is not a big factor in determining the communities, at least at the level of resolution that we're looking at. So we're finding very similar species in our communities, regardless of where the cheese is made. It seems to be mostly determined by that style of cheese, whether it's natural rind. So two natural rind cheeses made in very different parts of the world will look very similar. But we haven't looked deeper, like genetically, to see if, okay, maybe you have very similar species in France versus Vermont, but there might be regional differences in what their total metabolic pathways that they have, right? So that there could be regional differences still in the maybe like flavor production potential of different species depending on where they come from. Yeah.
Okay, so if I'm understanding this right, the bacteria and/or the fungi aren't coming in through the milk. They're coming from the environment and settling on the rind?
We think probably a combination of both.
Okay.
Yeah.
I can sort of continue on that then. The cheese curd process, if it's, say, cow's milk, is that pretty much uniform what bacteria are going to be on those types of cheeses, or is it the rind inoculation?
So we also don't see a big pattern. We don't see big differences within a similar style of cheese between cow, goat, and sheep. We see fairly similar types of organisms.
Cheese curd then itself doesn't make a huge difference, or is that affecting the taste just because of the proteins that are in the cheese curd?
Yeah, it's just because of the milk. Yeah, so the composition of the milk from those three different animals is different. The fats are different, which like sheep milk has very specific types of fats, which give it sheepy flavor, and there's goaty flavor.
It's going to be relatively similar though for all three.
The bacteria seem to be similar.
Yeah, I was just wondering, do they have to worry about contaminants like cheesemakers? Can like a whole batch be ruined and they have to start again? Or?
Yeah, so when cheese goes bad. So there are foodborne pathogens associated with cheese. So Listeria, actually Staph aureus, and E. coli can grow on cheese. It's fairly infrequent, but it does happen. And it's something that many cheesemakers will actively monitor every batch of cheese to make sure they're pathogen-free. And there's a lot of sanitation protocols during cheese making. I think I made cheese for a couple of months before starting my lab, and I've never done so much cleaning in my life. Yeah.